Trying to detect endangered aquatic species is often like trying to find a needle in a haystack, but underwater. It's a lot tougher. However, we need to sample these species so that we can determine their habitats and better protect them. So we use tools like nets or traps that can catch these fish. However, this becomes a problem when we're dealing with endangered species because these methods are often quite harmful. And the last thing we want to do is cause more stress to an already stressed species. Fortunately, there's an alternative tool known as environmental DNA, or eDNA. It's the DNA that exists outside of the individual's body and in its surrounding environment. So for aquatic habitats, that can be in the form of blood, shedding fish scales, urine, feces, all floating around in the water. For my master's, I wanted to test eDNA's strength in detecting a rare species in a natural system. I wanted to answer 1, could I even detect my species using just water? And 2, from a fixed point, how far downstream could I detect its DNA? I used Atlantic salmon as my target species as I was able to obtain Atlantic salmon water, which would be my eDNA source because it contains all of their cells. I let this water drip at a fixed point into Little River. This is a system which did not previously have any Atlantic salmon in it, hence any of their DNA entering the river would be coming only from my water source, and it's such a low amount to begin with that this would mimic a rare species. Now, I expected to only detect Atlantic salmon DNA maybe just 400 meters downstream, simply because this is a natural system. The water is constantly flowing, it's turning, dilution is happening, and there's DNA from hundreds of other organisms all in the mix. So to actually obtain eDNA samples, we begin with water collection. I took various water samples from my fixed point up to 1.2 kilometers downstream. These samples are filtered and all the debris, sediments, and DNA molecules are captured onto fine filters, which can then be broken up and extracted for your DNA. Finally, that DNA can be amplified and analyzed for your target species. So after this whole process, guess how far I was able to detect Atlantic salmon? Up to 1.2 kilometers downstream. This means that eDNA can be quite sensitive in detecting rare species, and if optimized, it can be cheaper, easier, faster, but ultimately, it's much more ethical because we're no longer fishing for the endangered fish to know if they're there or not. We can simply fish for their DNA. Thank you.